As Dragos mentioned previously, we will be talking today about the importance of animal models in translational medicine, while also focusing on the discoveries made by Arvid Carlsson. And I will be also getting into a few other studies which I believed were quite relevant to the topic. Before getting into the article, we first need to understand a little bit about the role of dopamine and its main action mechanism. First of all, it is part of the catecholamine family, together with norepinephrine and epinephrine. And some of the key roles of dopamine include motor control, motivation, reward, and reinforcement. But there's also sexual gratification. It can be involved in lactation and many other things. Dopamine acts through four major pathways in the brain. However, today I will be only getting into three of them, which are more relevant to our topic. The first one is the nigrostriatal pathway, which is highlighted in this picture with red. The neurons in this pathway originate in the substantia nigra pars compacta in the midbrain and then project to the dorsal striatum. And its main role is motor control. Then we have the mesolimbic pathway, which is highlighted in green. Its neurons originate in the ventral part of the tegmentum and project to the nucleus accumbens or the ventral striatum. And the main role of the nucleus accumbens is it is the center where reward processing happens. So this pathway is highly intertwined with the mechanism behind both psychosis and addiction. The third one is the mesocortical pathway. The neurons originate in the ventral part of the tegmentum and then project to the prefrontal cortex, and it has a very important role in cognitive function. Here I have summarized a little bit the exact locations for each one of them. As I've said, the nigrostriatal pathway originates in the substantia nigra and goes to the dorsal striatum. The mesolimbic in the ventral tegmental area and goes to the ventral striatum. And the mesocortical also in the ventral originates also in the ventral tegmental area and goes to the prefrontal cortex. All right, so how is dopamine produced in the brain? It first starts with the amino acid tyrosine, which is converted to DOPA by the tyrosine hydroxylase. DOPA is then converted to dopamine by DOPA decarboxylase. From there on, it can also be converted to norepinephrine and epinephrine, but we won't go into that too much. Dopamine mainly acts on two types of receptors, D1 receptors and D2 receptors. D1 receptors are part of the D1-like family, which include D1 and D5 receptors. They act on the GS protein, which activates adenylyl cyclase, which in turn increases the level of cAMP in the brain and activates protein kinase A. Among their functions, there are two major ones. Motor control, they act through the direct pathway in the basal ganglia. And a reduced activation in this pathway is found in Parkinson and causes symptoms like hypokinesia, because this pathway controls voluntary movement. It is also involved in reward and reinforcement through the mesolimbic pathway. And D2 receptors are part of the D2-like family, which include D2, D3, and D4. They activate GI or GO proteins, which inhibit adenylyl cyclase, lower the level of cAMP, and inhibit protein kinase A. Their functions also are involved with motor control. They act on the indirect pathway in the basal ganglia, which suppresses unwanted movements. And hyperactivation of this pathway is linked with rigidity and bradykinesia in Parkinson. It is also involved in psychosis and addiction, as I've mentioned previously, which are correlated with the mesolimbic pathway. It's actually, a lower level of D2 receptors is linked with a higher susceptibility for addiction. So, yeah. Okay, so to sum it up, the basal ganglia circuitry includes two major pathways. The direct one, which is responsible for movement initiation and starts with the D1 receptors, and the indirect pathway, which is responsible for movement suppression and is activated by D2 receptors. Therefore, dopamine deficiency will lead to a reduced activation of the direct pathway and an overactivity in the indirect pathway, which explains some of the major symptoms in Parkinson. So what is Parkinson's disease? It is caused by a neural loss in the substantia nigra pars compacta, which then translates to a lower level of dopamine in the basal ganglia. When symptoms of Parkinson's disease actually start to emerge, there is already an 80% loss of dopamine in the basal ganglia. It has an incidence of 5 to more than 13 new cases once in every 100,000 individuals. This varies a lot because of different types of populations, how different studies were conducted. But it is certain that it is twice more prevalent in men, with a little exception. In Japan, I found that it seems to be more prevalent in women than in men. And its incidence increases 5 to 10 times more from age 60 to 90. You can also see this in that graphic on the right side, how it goes high very fast after the age of 60. The two most major implications of this disease are neuronal loss in the substantia nigra, as I've mentioned, and also the presence of Lewy bodies in the neurons, which are aggregates of alpha-synuclein. I have so Parkinson's symptoms are split into two major categories. First, we have the motor symptoms, which are mainly bradykinesia, which is a difficulty with fine movement, tremor while the muscles are at rest, rigidity, blinking less often than usual, micrographia, which is very small writing. The person is unable to write as they normally would. Mask-like facial expression, which is also called hypomimia, which is determined by lack of abilities to control the facial muscles. Dysphagia, trouble swallowing, which then causes the drooling, and hypophonia, which is a softer voice than the person would usually have. Okay. Lewy bodies are thought to be involved with the cause for the non-motor symptoms. That's why I have included them here. As I've said, they're aggregated alpha-synuclein, which originally appear in the cholinergic and monoaminergic brainstem neurons, or in the neurons of the olfactory system. And then they move to the limbic and neocortical brain regions. This also correlates with the symptoms I will present after this slide. So in the right picture, you can see on the top image the difference in the mass of substantia nigra from the left picture to the right, where is the case of Parkinson's, with, which is the case of a person with Parkinson's. And in the middle lane, you can see how that very dense inclusion of alpha-synuclein distorts the body of the neuron and also the axons. On the bottom side, you can see in red the specific areas in which these bodies start to form. You can see they originally form in the brainstem and then move on to the basal ganglia and later to the rest of the cortex. Okay. So, the non-motor symptoms include cognitive impairment, autonomic dysfunction, sleep disorders, and hyposmia, which is an impaired sense of smell. I will also be covering the main mechanism in which different medications act on this deficiency. But first, we need to understand how the signal in dopaminergic synapses is actually ended. In the presynaptic neurons, there are some things called dopamine reuptake transporters, or for short DAT, which reabsorb dopamine after it is used and inside the presynaptic neuron, it is further metabolized by monoamine oxidase and catechol O-methyltransferase to other metabolites. Okay. So the main drug therapy that can be used in Parkinson's, our most important candidate is levodopa or L-DOPA, which is a precursor converted to dopamine, as we've seen in the first slides. This can be coupled with carbidopa, which inhibits the decarboxylation of dopamine in the of DOPA in the systemic circulation and allows for a better absorption of the central nervous system of this medication. Then we have MAOB inhibitors and COMT inhibitors, which will prolong dopamine activity, and dopamine agonists, which mimic dopamine effects. These have a moderate therapeutic effect, while the MAOB inhibitors and COMT inhibitors are mainly used for the initial phases of Parkinson's. Lastly, there is deep brain stimulation, which modulates basal ganglia circuitry in a systematic manner. However, this is a picture of deep brain stimulation. You can see those two antennas that go very deep into the brain. The things on the bottom side are not related to this procedure. So this is a technical procedure in which two neurostimulators are implanted deep into the brain, and they're able to send electrical signals to specific targets inside of the brain, which correlate with different disorders. So this treatment can be used for movement disorders like Parkinson's disease, essential tremor, and dystonia. Neuropsychiatric conditions like obsessive compulsive disorder or OCD, and even neurological disorders such as epilepsy. An interesting thing about this is that the mechanism for why it works is not entirely known, although it is believed that it works mainly by suppressing pathways that are usually overstimulated in these specific conditions. Okay. Finally, we will be getting into the article, which I suppose you have all read already. Okay, so it is important to notice that at the time when this article came out, which was in 1957, when the original discovery was made, most scientists believed that the majority of synapses in the brain and the central nervous system were electrical in nature. And this discovery actually paved the way to understand that most synapses are actually chemical, not electric. Okay, so Carlsson wanted to see whether he could revert the symptoms of reserpine on these bunnies by using noradrenaline. Reserpine is a catecholamine-depleting drug, so it will get rid of noradrenaline, dopamine, and everything related to this family. It was believed at the time that noradrenaline was the main neurotransmitter responsible for movement control. And he believed that by treating these rabbits with L-DOPA, that they would regain their noradrenaline levels and they would become normal again. What he didn't expect, however, is that L-DOPA still had a very benefic effect on them. Like you can see in the picture, they went from akinesia, being unable to move, and being almost sedated by this drug-induced depletion of dopamine, to the bottom picture where they were again seeming very active and they had regained most of their motor control. This happened because well, after the treatment, their noradrenaline levels were still depleted. They had an elevated level of dopamine, and this allowed them to regain their normal functions. From this, he realized that Parkinson's disease is the result of a dopamine deficiency rather than a noradrenaline deficiency. And he was also able to formulate later through the study of dopamine, what is now known as the hypothesis of the hypothesis of schizophrenia. So he basically was able to, for the first time, understand what happened to patients who were schizophrenic. Despite this major discovery, it took another like 10 years for L-DOPA to become the standard treatment for Parkinson's disease. Okay, so first I want to mention a few things about schizophrenia. The main thing patients have to struggle with is psychosis, which can manifest through hallucinations, most often auditory in nature, or delusions. The difference between these is that hallucinations are false perception of an external stimuli that doesn't actually exist, while a delusion is a fixed false belief that you cannot challenge that person upon. It has a few types of category for symptoms, but the most important ones are the first three. We have the positive symptoms, which are the psychotic symptoms. We have negative symptoms, which represent a decrease in emotional range, poverty of speech, loss of interest and drive. And we also have cognitive symptoms like neurocognitive deficits, for example, an attention deficit. And lastly, there are mood symptoms, which are kind of, they make sense, taking in consideration all of the rest, that their mood also wouldn't be exactly fine. All right, so what did Arvid Carlsson discover? While working with chlorpromazine, which is an antipsychotic medication, he realized that antipsychotic medications worked by blocking the receptors for dopamine and partly receptors for noradrenaline and serotonin. That's how he took a great part in forming what is now known as the dopamine hypothesis of schizophrenia, which basically explains the fact that in the case of schizophrenia, there's a hyperactivity in the mesolimbic pathway. Again, the one in green on the right picture, which correlates with positive symptoms like hallucinations. And there's a dysfunction of the mesocortical pathway, which will influence the negative and cognitive symptoms of the illness. I've also wanted to include a few other implications for different diseases which highly involve dopamine. Firstly, we have depression, in which the anhedonia found in this disease is linked with low mesolimbic dopamine. Anhedonia basically means an inability to experience joy or pleasure with things. Then we have addiction, which is correlated with that reward pathway I mentioned previously. The mesolimbic dopamine is heightened as a response to using drugs, which then reinforces the need for continuous use. And in the chronic use, there's a receptor downregulation, which turns to tolerance. In ADHD, there's also dopamine dysregulation, which is localized in the prefrontal cortex and causes inattention, as the prefrontal cortex is correlated with different cognitive functions. Then we have the Tourette syndrome, in which we have an excess of dopamine in the basal ganglia, which causes tics. And the treatments, interestingly enough, includes antipsychotics because they inhibit those dopamine receptors. There's also restless leg syndrome, which is treated using dopamine agonists, and Huntington's disease, which is also a dopamine dysregulation. Okay, so now we will be talking about a few other studies which have used animal models to prove very important things in neuroscience. First, we have this one, a primate model of Parkinsonism, which was published around 1983, if I remember correctly, by a team of scientists in Maryland, where they treated monkeys with this compound with a very long name, which I will be calling MPTP. Yeah, so the original story behind this study is that in the 80s, there was a series of patients who presented with drug-induced Parkinsonism after they injected themselves with an illicit narcotic drug. And this team of researchers wanted to find out whether the compound responsible for this was an MPTP, because they were uncertain. After treating these monkeys for around 5 to 8 days, these are some of the symptoms that they found. These acute symptoms usually occurred after the third time of exposure to the drug. And initially they lasted for 15 to 30 minutes and occurred after 5 minutes of each injection. The acute symptoms were intermittent eyelid closure, loss of facial expression, and postural tremor. As you can already notice, they kind of resemble the most common symptoms of Parkinson. And eventually they became chronic after around 5 days. They gained general slowness of movement, of flexed posture, loss of hand dexterity, and they also had these so-called freezing periods, which are showed in the picture on the right with the monkey who was totally flexed, its back arched, and the limbs were also hyperflexed. The picture on the left is the same monkey after receiving L-DOPA treatment, which was described as being more energetic than usual and having regained all of its motor control. Some of the neurological findings that they were able to make were also that after sorry. After dissecting the brains of these monkeys 27 days after receiving treatment, they realized that only half of the normal cells were still present there. And they analyzed their brain using histofluorescence. However, I couldn't really find the picture of that here to post. So we'll have to use our imagination a little bit. They also noticed the presence of swollen axons and a different form of neuronal bodies. And these changes mainly occurred in the ventral part of the internal capsule, the basal portion of the globus pallidus, and the medial part of the putamen. I've included this picture again to show you kind of how those axons would be distorted. And you see on the left upper picture the size of that inclusion, how much it can alter the shape and function of that neuron. Okay, so what they found could be summed up into these three things. First, they realized that the levels of HVA, MHPG, and 5-HIA had fallen to an average of 37%, 38%, and respectively 60% of their baseline. What are these things? HVA is homovalenic acid, which is a metabolite of dopamine. MHPG is 3-methoxy-4-hydroxyphenyl glycol, not that important. It is a metabolite of noradrenaline in the brain, and 5-HIA is a metabolite of serotonin. So they could accurately measure the effect of this drug on the brain. They also concluded that obviously NMPTP was the neurotoxic agent responsible for that drug-induced Parkinsonism in humans. And they suggested that the NMPTP-treated monkey could be a possible animal model for treating different therapies for Parkinsonism. And some people really tried that. This is another article which was posted in the Journal of Neuroscience in 2023. It's quite recent. And it basically wanted to measure how... Actually, I need to show you this picture first because it's quite an interesting one. It was included in the study. So these monkeys were treated with the same compound. And after they exhibited the symptoms for Parkinsonism, they were strapped to this chair. And on the left arm holder, there was also a mechanical oscillator, which could induce some passive rotation of that limb. And in the central axis of that device was a goniometer, which basically measured the amount of the exact position at in each second of that arm. So basically, it measured the rotation. And there were also strapped on each limb with 3D accelerometers, which recorded their movements. They had a recording chamber implanted into the right side of the skull to be able to get online neural recordings. I suppose it was the right side of the brain, although I couldn't find in the article because they were most interested in the left arm of the monkey, and those signals would cross over to the other side. Okay. Okay, I have a little bit more to talk about this. So, in this study, they basically wanted to see if by inducing these passive rotational movements, the neurons that were affected in Parkinson's would respond differently, and they were able to tell that they were more activated than usual. However, I don't know if this could actually lead to some kind of therapy in the future. It is most important to understanding the mechanism behind it. The last study that we will talk about is this one that was published in Nature in 2017. It was led by Jun Takahashi, if I say his name right. And it wanted to see how induced pluripotent stem cells could be a possible treatment for Parkinson's. And they tested it also on monkeys, as you can see in that picture. So what are these iPS cells? Induced pluripotent stem cells were obtained in this experiment by using neurons both from healthy monkeys and monkeys with induced Parkinsonism, and reverting them to pluripotent stem cells to be able to be implanted into their brain. The most important thing they found was that these monkeys not only survived for 2 years after this, but they also had regained a lot of their motor control after having this procedure, which shows that these cells were able to form connections with their neurons. And another two very important aspects which usually makes people refrain from this kind of research is the chance for these cells to become tumors in the future. But they were able to show that these cells didn't show any signs of possibly becoming tumors. And also that they were not immune to immunosuppressants. So they could be, I don't know how to say it, they could be killed basically if they ever wanted to, without affecting the person that had them implanted. Okay, so that was all. I hope you found it informative. And I hope this highlights a little bit the potential use we have for animal models in science. Thank you.
Thank you. I think one thing that is important to mention here is that Arvid Carlsson's work earned him a Nobel Prize in 2000 for his work on dopamine, because prior to his work in the 1950s and 60s, it was thought that dopamine was just a precursor for noradrenaline. Noradrenaline was the big neurotransmitter at the time, and it was thought that dopamine was just a precursor, and his work, and it was animal work, showed that dopamine was a neurotransmitter in its own right. It's kind of a success story in terms of translation, being that the original work was done on animals, but much less so in psychiatry, where, and this is a story that is valid for all psychiatric disorders, we kind of went the other way and we kind of reversed engineered what we understand about dopamine, because we first found drugs that had clinical effects. So we found chlorpromazine to actually reduce psychotic symptoms, and we had no idea why. And then after we started treating patients in an era where we didn't really need to do randomized controlled trials, we could just introduce it in the clinic, we started to kind of reverse engineer what was about chlorpromazine that would be antipsychotic. And then we later figured out that it was working on dopamine systems. With Parkinson, much less so, it was more translational. They were able to actually go in and look at animal models. And that is because Parkinson is or at least motor symptoms of Parkinson are significantly simpler than psychotic symptoms, and they are much more amenable to animal models. That's one of the big differences, because we were talking with Dragos, like, what is the difference between schizophrenia and Parkinson in terms of translational qualities, and why we have so little work of translational qualities schizophrenia and psychiatry in general, as opposed to Parkinson. So I think that's the key takeaway here, that Carlsson earned a Nobel Prize in 2000 for his work on dopamine, because he actually discovered dopamine as a neurotransmitter.
Yeah, that's some very, very good insight. And because I think that, again, we're talking about a study that actually translated, and studies that actually have more translation room than others. And always when talking about animal studies, it's very, very tricky with this translation part. Um, that's what we were, as Barbu said, it's what we were talking about earlier, just before the journal club started. And well, if you are to actually go into why these studies translated whereas others didn't, you would have like a semester worth of journal club. It's a very, very long discussion. But part of it, maybe just because it's a very, very clear pathophysiological mechanism, like unlike, let's say, autism, where you have a very multifactorial disease. Here you have a few factors with a very, very big impact, whereas otherwise you would have a lot of factors with little impact that work together to create the disease. So here it makes it much easier to find them. And as we're saying, we're a bit unlucky, because if you wanted to do big discoveries, well, they've been taken usually. The things that are easy to discover have already been discovered. But something else that we may look to, I think, is again, all no models are good, and there are only like less bad models, and you try to get the least bad that you can for your purposes. And a lot of the studies that you cited were on primates. So that's something that, of course, will help in any sort of study that you're doing. They're much closer to humans than, like mice are, or even if you want to be more far removed, zebrafish or Drosophila or so on. So, but at the same time, they're very expensive studies. They're studies that are very, very hard to do. I'm not even sure that if we have primates in Romania that we're like in labs. I don't. They're very expensive, and besides expensive, it's yeah, the ethical aspect of it is very, very hard to actually get done. It's much easier for you to get a permit to work with fruit flies than it is with monkey, which is so much similar to us. And that's why a lot of studies actually don't only call them primates, but call them non-human primates, to remind us that we are primates as well. So much closer, much easier to translate. Any thoughts on it? If any of you managed to read through, yeah. Hopefully right now.
Yeah, I have. Okay, so I wanted to add just one thing. I think they are probably not the best models in order to study the basic mechanism behind certain drugs in humans, but they can certainly be useful in finding the right dosage that would not kill a human before actually trying it on them, you know?
Of course, well, I'm not sure.
Okay, sorry.
It's too expensive to use it as a
when we're
study.
when we're talking about what you're saying, like when we're talking about toxicity, I think that a lot of times it's actually easier to do like more high throughput methods, models like the zebrafish, for example. Again, that's when you're looking at things that are a bit more similar in all living cells, like toxicity. Of course, they do differ from species to species, but you would prefer something that you can do a lot of testing, that you can have thousands of zebrafish. They're very easy to reproduce, so it's easy to actually know that you find good results, whereas you wouldn't really, it's very expensive to test it on thousands of monkeys, or other primates. So for that, not so much. I think that where these models shine is in more uh, fundamental parts. If you're looking to things that you want to translate later more towards human, they may be more similar, than and again, descriptive studies, or if you want to do, like some late stage studies before entering, human trials. Even though at least in Europe, I think that animal studies have been removed as a condition to start human trials. Yeah, yeah. I think that they've, yeah. But one of the studies that you cited was, I found pretty interesting, the one with the monkeys, the one with the iPS cells in monkeys. They were immunocompromised monkeys, right?
Okay, so initially, yeah, they were immunocompromised. That is a given requirement for implantation of this type of cells. But they also found that they could survive in their body after some time without the need for immunosuppressants. So, yeah, I think it's still a pretty good find. And also, I was able to see that there was also a study, or at least a case, in Japan, with a woman who had retinal, who had some cells of retina implanted into her eye. I don't know how to say it. So some of her cells were reverted to pluripotent stem cells, and then they were able to be implanted into her retina to restore some of her vision.
Why I cover that is because, yeah, so induced pluripotent stem cells are, as name says, induced. So you would take usually a bit of skin, you take out the fibroblasts, you turn them into pluripotent stem cells, and then you differentiate them into whatever you want. But of course, if it's not from the same patient, which you usually would use them when you're talking about humans, but here it was human iPSs in monkeys, um, you would usually have an immune response and they would be killed off pretty quickly. Uh, but here it's very interesting that they survived for a bit. But again, that just goes to show immunocompromised mice are insanely expensive and they require different facility than any other. These mice, they cannot fight against disease. So all the food that you give them has to be sterilized before, all the bedding, everything, and you have to be completely sure that you are as sterile as you can, because any infection will be deadly to them. They have no way of defending themselves, and that's mice. And they're much easier to reproduce, they're much easier to work with, and they require less space and less resources. So immunocompromised primates would be an insanely expensive model, and I think that there are very few facilities in the whole world that could accommodate such a model. That's why I found it interesting. Did they? I know that you said that they could survive for a bit, but could they survive enough to actually induce an effect on the disease?
So, as I've said, in the article, they were able to survive for about 2 years, and they also were able to regain some of their motor control, which showed that those cells were at least a bit. Yes. Honestly, I'm not very sure, but it might be the case, as you mentioned, that the immunocompromised monkeys are not a very reliable model for a lot of types of experiments. But one idea that I found to be interesting of coming from the author was that in the future, in the hopes of implementing this type of therapy for actual Parkinson's patients, he wanted to form some cell lines from all kinds of different patients, to be able to find a closer genetic match for future Parkinson patients who could have this implemented on them. And I think it's quite an interesting idea. I don't know if it's very doable, but yeah.
So the magic of induced pluripotent stem cells is a bit in the fact that you can make them from your own patient. Cell lines, don't get me wrong, are very good to research, but if you want to implant them in patients, then you need to actually also give that patients immunosuppressants, because otherwise they will kill them. And that comes with a lot of other risks, which, like very similar to transplantation. You would have a patient that basically you want to keep immunocompromised for as long as you can, because otherwise, again, the cells would be killed. And something else is, as you mentioned, the danger of induced pluripotent stem cells of turning malignant, and if not malignant, to something else. Because if you've worked with IPCs before, they are very unstable. They're very volatile. Even changes in temperature can make them differentiate into something weird. And even when you're making cultures, like from differentiating them, you don't call them neurons, you call them neuron-like cells, because they're, not actually neuron state. When it comes to neurons, of course, depends on the cells, but again, they can also, if you don't control the parameters right, they can also turn back into stem cells. And the human body is something that well, you can't really control the parameters in the human body. So you can't really control if they stay like that or not. And you don't have those all these differentiation parameters in an adult body as you would have in an embryo. So that's why you don't really see stem cell treatments as part of any working therapies, because you have these problems. And again, very nice, very impressive that the author didn't find any malignant transformation, but since it's a model that is so expensive, it's very hard to replicate, so and to reproduce. So nobody, very few people would be able to test his theory and to say, yes, I can find that there is no malignant transformation. So that's the problem with because you mentioned that they may not be reliable. It's not, they are reliable. They are easier to translate, but they're harder to reproduce. So So it's hard to actually know if the data that they present is reliable, and if the data is actually true, or if it's just either false positives or somehow influenced. And so yeah, something that about this that I've mentioned in previous journal clubs and presentations and so on this topic is it's very different, like for a researcher and a surgeon. A surgeon likes to have very low reproducibility, like it's very nice to say that no one else can do the surgery. But if you as a researcher say that no one else can get these results doing what I'm doing, that's not really that big of a flex. So that's why models are important, but it's a very, very interesting study, nevertheless. Do you have any other comments?
So just so we don't get lost in the details about the translational qualities of these studies, the actual translational success here is not only Arvid Carlsson translating his his fundamental work on dopamine, understanding dopamine. It is the initial treatment for Parkinson that was induced experimentally by Carlsson in rabbits in the 1950s. By giving them reserpine and creating severe motor deficits and then reversing them with L-DOPA. And that's one of the hugely successful translational stories for clinical medicine because it's translated from animals to humans. More recent studies, yeah, there's a lot of confounds, there's a lot of trouble with them. But this is one of the highly successful stories of translation, I think. So this is one of the key takeaways. What did you guys think about the article? It's kind of a narrative article. It's kind of a looking back after he won the Nobel Prize. It's 1 year after he had won the Nobel Prize, so it's a little more narrative. But I wonder what you guys thought about it. Alex, what did you think about it?
Well, honestly, I thought it was really like an inspiring thing, seeing like a line of studies that actually translate into humans and like basically a very basic hypothesis turned into the discovery of Parkinson's and many other pathologies, as Laura said. And also, I found it very interesting. I found the fact that honestly, before like I got into more sciencey part of medicine, I didn't hear about any other models except mice. But seeing, even Carlsson used rabbits and monkeys and stuff like that, I think it's very interesting, and I think it's like a train of thought to think like what other models could be used to research stuff. Maybe mice aren't the only answer. Any other thoughts?
So I also found the research interesting, the article research interesting, but my question would be, what can Carlsson's work teach us about the future of translational medicine, translational research? Because I don't know, my question is, are we still dependent on animal studies in order to make such breakthrough discoveries, or the new technologies have reduced this need of animal studies, like, I don't know, stem cells, organoids, or, yeah.
That's a very good question. I think that people are very much fighting over that question in the whole science world. I think that at least what the EU thinks is that we've moved past the need for animal studies in general, and that of course, there are some places for it, but we're trying to move past them, and we're seeing that again. This was also, we have to keep this in mind, this was also his luck that it wasn't found yet, that it's something that's pretty easy to find, because it's just, like one condition that causes such a big effect. And as I mentioned, I think that we've found most of these easy to spot things. And right now, moving on to more complex pathologies, it becomes more and more dependent on the species, and we can feel where these animal studies, like are actually falling off, and they're starting to get further and further from the truth. So I also am of the opinion that for a lot of the clinical aspects and for a lot of the things that are going to translate more, not saying anything about the fundamental part, these in vitro models that try to replicate the human body and the human environment, they're going to be much better in, yeah, but I'm not sure if anyone is of the other opinion. Yeah.
I don't really disagree with you. However, just wanted to ask to add something. You've mentioned that Parkinson seems like a simple disease, but despite having this type of mechanism explained behind it, we still don't exactly know what is behind the formation of the Lewy bodies, or what actually causes the loss of dopaminergic neurons. And I have found that there are very many different plausible causes for it, including different types of Parkinson, which can be linked either to genetic changes or to metabolical changes, So as you said, I think that the research that has to be conducted today is so complex that animal models probably can't replicate closely enough the conditions we see in humans. And I also think that organoids and other studies on cells probably also can replicate the exact conditions that would have to be met. So, no, that's all, I think.
No, as I said, I really like that phrase that there are really no good models, only less bad ones. But yeah, it's true. It's, of course, I'm saying that it's simpler because even right now we're not talking about that much about, like, what causes the symptoms, because we know that it's this lack of dopamine. We're looking into what causes the lack in the first place, of course. Just saying that very, like, lack of dopamine was simpler to find. It was something much more obvious than more multifactorial diseases such as autism, such as cancer formation, a lot of them that are not really linked to one specific gene and so on. And of course, you can't really mimic everything with in vitro models like you can't with any other model. But yeah, that's, I think, a good observation. That's some of the limits that we're facing right now, I guess. Okay.
I think the 1950s are ripe with discoveries of neurotransmitters. John Eccles was the one who got the Nobel Prize for settling that debate, whether synapses are chemical or electrical. And once they had settled it, it was a very productive period in the 1950s and 60s for people discovering neurotransmitters. You see serotonin, Bernard Brody. You see there's a few other neurotransmitters that were discovered there, and Otto Loewi had discovered the first one in 1920s and 30s. But with regard to translational animal models, there's one more aspect with regard to schizophrenia and all mental disorders, is that animal models are really not that good, because you don't really know if you've created the symptoms that are verbally reported by patients. Mental disorders are usually highly correlated with people reporting that they have those disorders. You can see depression-like or schizophrenia-like behaviors, but we don't really know what exactly goes on in the mind of a rat that is part of an animal model of schizophrenia. So with regard to that, Parkinson's is a simpler disease to work on, because you can produce motor symptoms and you can actually observe them without having the need of the patient to report the symptoms. So that's another critical setback that animal models have.
Yeah, but at the same time, as Dragos said, in vitro models are better for like simple pathologies, but like for mental disorders, I think they're much, much worse. Like, yeah, that's what I wanted to add.
Of course.
You can't tell like that a cell is depressed or schizophrenic.
Um, yeah, I agree. I think that for in vitro models there, it's like it depends on how much the genes play a part of it, because you can't tell if a mouse is hallucinating. You can tell much less about whether an organoid is hallucinating, you know. Um, but again, something I found very funny is that we're actually very, very skilled at treating mice. It's just that we can't really treat, you know, like humans, unfortunately. And I think that with a lot of these models, we also have to be careful not to, you know, like get into selection bias or like confirmation bias, because a lot of times when we create models, we uh, start by thinking about how it looks. We do something that works in the way that we think something works, and then we study it, and so we can get different confirmations, you know. Like, of course, here it's pretty easy, but if you look at, you know, like certain gene knockouts, for example, of course you're going to find that stuff related to that gene, not necessarily to how the pathology works in patients, you know. So you don't know for sure that it's that accurate of a model, and again, a lot of limitations. Any questions? Um, if not, I hope, or any other comments, anything else? Otherwise, I really hope that you had a nice time, and I hope that you felt a bit more inspired by something that actually worked, because again, we tend to go into why studies don't tend to work. I think that especially because we are a bit conditioned to believe everything, it's very important to know when to not take things for granted. But I also think that it's important to see sometimes, you know, like an example of successes, not only failures. And I hope that at least it may have sparked your interest to read more into either Parkinson or these sort of animal studies, and that you may have found out more about the limitations that you may have in animal studies. Maybe start a monkey lab in Romania. Yeah, a non-human primate lab. Yeah, that would be great. We're also, maybe you've heard, we're also part of, the university as a whole is also part of larger consortium, Neurotech EU, and we as a student circle, we basically represent the synapses group, the student group of Neurotech in UMF. And for this, they're trying to get some feedback over how the events are working at each university. So we would really appreciate if you could answer that feedback form when you have time. It doesn't really take a lot of time. It's very quick. We'll send it in Teams, like as a reply to this meeting. So you can find it there. Again, please, it takes just a few minutes. We would really appreciate it if you could do it. So with this, thank you again, and I hope to see you at our next edition.